An overview of fungal identification, methods, and strategies. So fungi collection and transport. So the importance of proper collection and transport helps to ensure accurate identification and diagnosis, preserves the viability and morphology of fungal specimens, and prevents contamination or overgrowth by other microbes. For collection guidelines, we're going to use sterile tools to collect the fungal samples and collect sufficient material, include parts with active growth or infection. We're going to try to avoid contamination with soil, debris, or other microorganisms. And then it's going to be transported to the laboratory. For transport to the laboratory, we want this to happen as soon as possible. Fungal samples should reach the lab as quickly as possible to help maintain their viability. Any delays can cause loss of viability, changes in morphology, and overgrowth of contaminants. We need to make sure we use appropriate containers, such as sterile, leak-proof containers. Paper envelopes or bags are preferred over plastic to avoid moisture buildup, which can actually cause fungal overgrowth or decay. Processing upon receipt, so samples should be processed promptly. Delays in processing can affect culture results and diagnostic accuracy. If immediate processing is not possible, we're going to store those samples properly, so in a cool temperature, but avoid freezing unless specific protocols require it. Primary plating media for fungi are essential for isolating and growing fungal organisms from clinical or environmental samples. Some media are formulated with antibiotics such as chloramphenicol and gentamicin, which inhibit bacterial growth and prevent contamination that could otherwise overshadow fungal colonies. Common media used for fungal culture include Soberdar's dextrose agar, or SDA, inhibitory mold agar, or IMA, and brain heart infusion agar, or BHI. So SDA agar has an acidic pH that favors fungal growth while partially inhibiting bacteria, and it is often supplemented with antibiotics for better selectivity. Inhibitory mold agar contains antibiotics specific to suppress bacterial contaminants and promote mold growth. Brain heart infusion agar is a nutrient-rich medium that supports fastidious fungi and can also be supplemented with antibiotics to reduce bacterial interference. For specialized purposes, potato dextrose agar, or PDA, is frequently used for slide cultures to study fungal morphology and sporulation, while cornmeal agar is preferred for yeast identification because it encourages the development of characteristic structures in Candida species. So using the appropriate media with or without antibiotics is crucial to ensure successful fungal isolation and accurate identification. So how do we separate molds from yeast? It's all about the temperature at which they grow. So molds grow best at room temperature, where yeast are best at body temp, so 37 degrees to 35 to 37 degrees Celsius. We call molds, some are dimorphic, so basically it grows in two phases. So it can be a mold at room temperature and then transition to a yeast at body temperature. And all of these will grow well in ambient air. One of the things that we can do for a quick determination of the presence of fungal elements is a KOH direct exam. So the KOH is a potassium hydroxide, and that will clear the cellular debris in the sample. Fungi will inhibit skin, nails, hair, and sometimes even sputum. So you may get a skin scraping, you can get a piece of hair or a piece of nail, samples. So we can do a KOH on that. It will disintegrate the debris, and then the hope is that you can microscopically determine the presence of any fungal elements in that sample. Cryptococcus neoformans is a yeast, and we use a negative stain called India ink. For Cryptococcus neoformans, usually your specimen of choice is going to be cerebral spinal fluid. So we have a dark background with a clearing and a halo around the fungal element when you have that type of specimen. You could also use fluorescent stains. We can also use gram stains. A tease preparation involves placing a small amount of fungal material in a drop of lactophenyl blue cotton stain, which helps highlight the fungal structures for microscopic examination. The Scotch tape preparation method consists of pressing a piece of clear tape onto a mold colony, then placing the tape sticky side down on a microscope slide for observation. Slide culture is a technique where the fungus is allowed to grow directly on a small agar block on a slide covered with a cover slip, enabling the study of fungal morphology and sporulation in situ. For yeast identification, the germ tube test is commonly used. It detects the formation of germ tubes, which are outgrowths from yeast cells that help differentiate certain species such as Candida albicans.